Yes, Cynthia, please.
Okay, I'm gonna give a little introduction of Dr. Malone. Um, it's for me a great pleasure to introduce Dr. Malone. Um, el doctor Jacob Malone es egresado de la Universidad de Oxford en el Reino Unido, en donde obtuvo el doctorado en microbiología molecular en el año 2005, en el laboratorio del doctor Paul Rainey, en el Departamento de Plant Sciences. Posteriormente, del 2005 al 2011, el doctor Jacob Malone realizó una estancia postdoctoral en el laboratorio de Urs Genal en Basel University en Suiza, y desde el 2011 se incorporó al John Innes Center en Norwich, en Reino Unido. Actualmente es profesor y jefe del Departamento de Microbiología Molecular en este instituto. Ha publicado más de 30 artículos científicos en revistas de alto impacto como Nature Microbiology, PLOS Genetics o PLOS Pathogen. Pues sin más, los dejo con el doctor Malone y su plática acerca del Control of mRNA Translation by Dynamic Ribosome Modification. Dr. Jacob, thank you so much for accepting our invitation so you can
It's a pleasure.
start this seminar. Thank you.
Thank you very much for the invitation. I'm going to, I've noticed now that my title has changed slightly, but the subject of my talk hasn't. I will be talking about bacterial control of mRNA translation by second messengers by ribosomal modification. And I'd like to set this in the context of plant microbe interactions, which is the wider area that my group studies at the John Innes Centre. So the John Innes Centre is the UK Government Research Centre for Plant and Microbial Science. So it's a fantastic place to work, and we get to do both applied and fundamental science. But in my case, it's to do with the interactions between plants and different microbes. So what I'm interested in my lab is bacterial decision-making during plant colonization. So I'm interested in interactions between gram-negative bacteria, specifically Pseudomonas, with both in both pathogenic contexts and in beneficial. So the main model organisms that we use are Pseudomonas syringae for infections and Pseudomonas fluorescens, which we use to study the colonization of plant roots. So these processes, it's easy to imagine them being sort of fairly passive. Bacteria just are on plants. But these are actually incredibly complex and directed processes. So the process of a bacterium infecting a plant or colonizing a plant root are both complex and directed, and also have multiple things in common. So you will find biofilm formation at different stages on the surface of roots or on plant surfaces, on plant leaf surfaces, I should say. You will find circumstances where bacteria undergo directed migration. So they will migrate across leaf surfaces into the apoplast in the case of an infection, or they will migrate through the soil towards the plant root, following a gradient of root exudates. You will also find bacteria will control processes to do with apoplastic proliferation, nutrient acquisition, and proliferation in the rhizosphere. And all of these processes need to be controlled. The bacteria will take in signals from its environment. It will say, what is the temperature? What's the, is there a plant nearby? How many other bacteria are nearby? What sort of nutrient conditions am I living in? And it will change its behavior accordingly. And so what my lab studies is how these different processes take place in the context of plant colonization. And the system I'm going to talk about today is a 3-gene operon from the plant growth promoting bacterium Pseudomonas fluorescens SBW25. And this, I think, is a fascinating system. We've been working on it for several years, and it controls how this plant growth promoting bacterium colonizes different plants. So the main model organism that we use for colonization experiments is wheat. And what we can say is that the Rim-ABK system is important for colonizing the wheat rhizosphere by Pseudomonas fluorescens. So let's have a look at the Rim-ABK system. So it's a 3-gene operon containing these 3 quite small genes. The first of them, Rim-A, encodes a predicted cyclic di-GMP phosphodiesterase. So this is a protein that's predicted to degrade the second messenger signaling molecule cyclic di-GMP. The second protein at the beginning of this study was just a protein of unknown function. We know a lot more about it now, and I'll talk a bit about that in the course of this talk. And the third protein was this protein Rim-K, which is a ribosomal modification protein, or is predicted to be. And the reason we started looking at this operon was in a 2009 paper from Chris Thomas' lab in Birmingham, they showed that this system, among other genes, was upregulated in the plant rhizosphere. We thought, well, okay, so why would you have an interesting little set of genes like this with this phosphodiesterase and this ribosomal modification system? Why would they be upregulated in the rhizosphere of a plant? So first question we need to ask is, okay, well, are these genes important for plant colonization? So we made deletions of each gene, and then we carried out competitive rhizosphere colonization experiments against a lac-Z labeled, so a blue labeled wild type bacterium. And we looked at the relative ability of these different mutants to colonize the plant root. And what we saw was this. If you plot your competition data for plants that have been colonized for 7 days by both your blue wild type bacterium and your white, so lac-Z minus mutant bacterium, and you look at the ratio of mutant to wild type bacterium that you can recover from the plant rhizosphere after 7 days inoculation, what you find is that both the Rim-A and Rim-K mutants are significantly compromised for the ability to colonize the wheat root rhizosphere. So what this means is that these Rim genes are important for colonizing wheat roots. So what might they be doing? Well, to answer this, the first thing we need to do is to find out exactly what these different proteins do. We'll start off with Rim-K. Oh, actually, sorry, what I should say is, it looks like these slides have been mixed up, sorry. It looks like the whole Rim system is upregulated in the early stages of rhizosphere colonization and downregulated in the later stages. So we know that the system is important, and we also know that the system is turned on and turned off throughout the process of rhizosphere colonization. So the question then is, what is the Rim system doing? And we'll start by looking at Rim-K. So the reason Rim-K was called Rim-K is because there is an E. coli homologue of this protein, and it stands for ribosomal modification protein K. The E. coli homologue modifies the ribosomal protein RPSF. And this is RPSF here. It's on the small ribosomal subunit, and it's ribosomal protein S6. And what the E. coli protein does is it adds chains of glutamate to the C terminus of this ribosomal protein. So we said, well, you know, is this the case? Is this what happens with our Pseudomonas fluorescens protein? So the way that we studied this was we purified Rim-K, and first thing we wanted to know is, does it function as an ATPase, which is what the E. coli protein does. So this is the reaction that the E. coli protein does. You have your ribosomal protein plus ATP plus alpha L-glutamate, and Rim-K catalyzes the addition of glutamate and the synthesis of ADP. So we can look at the ATPase activity of our purified Rim-K, and what we find is that the protein is an ATPase, and also that ATPase activity, as you can see in these two top curves here, where we have glutamate added, the ATPase activity is stimulated quite strongly by the addition of glutamate. So it looks like we have a glutamate-dependent ATPase. And what we can also do is purify RPSF. So we now have RPSF here and a small amount of RIMK here. And when we incubate the two together in the presence of ATP and increasing amounts of glutamate, what you find is that RPSF goes from here, it gets bigger and bigger. And we've actually tested using mass spec. We can show that these bands contain an RPSF protein and then an extremely long chain of glutamates at its C terminus. So thousands of glutamates are being added. So it looks like we do indeed have an alpha-L-glutamyl ligase which targets the protein RPSF. So this protein is functioning like the E. coli protein. And we said, well, what does that actually do? If you have glutamyl ligase activity, if you are modifying your ribosomes, then, what does it do to the cell? And the way that we answered this question was to conduct a proteomic analysis. So we made a deletion, or we took our deletion of RIMK and we grew those cells overnight, and then we looked at the proteins that were present in the soluble proteome of the RIMK mutant and of wild type. And what we found was that a series of proteins were downregulated in our delta RIMK strain. And specifically, we found a huge number of ribosomally associated proteins were downregulated. But when we looked at the upregulated proteins, that's where things got really interesting, because the upregulated proteins included all sorts of things that we would associate with rhizosphere colonization. So we found things like ABC transporter components for amino acid transporters. We found things like pyoverdin biosynthesis genes. We found stress response genes. And overall, we found a series of things that we would associate with colonizing the rhizosphere. And what was also really interesting was one of the downregulated proteins was a translational regulator called HFQ. And HFQ is associated with the regulation of all of these genes. So we were able to put together kind of a simple model at this stage for how the RIM system might be functioning. And we published this in 2016, and this was the model that we published back then. This model suggests that in the early stages of rhizosphere colonization, when you have an increased expression of RIMK, you'll see increased RPSF glutamation. That leads to increased abundance of HFQ, which leads to suppression of various RNAs, and ultimately it leads to the promotion of motility, virulence in pathogenic strains, and also metabolic adaptation towards glycolysis. In the later stages of rhizosphere adaptation, and I should say we've also examined this system in the pathogen Pseudomonas syringae, and it seems to work pretty much exactly the same way in syringae. So what we say for rhizosphere colonization is also true for infection. But in the later stages of rhizosphere colonization, you see reduced levels of RIMK activity. This leads ultimately to less HFQ, an increase in HFQ mediated or regulated mRNAs, and this leads to processes including root attachment, metal ion acquisition, oxidative stress responses, and organic acid transport using ABC components, ABC transporters. So this was a nice, simple model. Well, it's quite a complex model, but you see what I mean? There's no mechanistic understanding at this level, but it's a nice model for what may be going on in the rhizosphere. But it's fundamentally unsatisfying because it doesn't really tell us what's going on. So to begin with, how is RIMK being regulated? We know it's being regulated by expression, or we know the expression changes, but we don't know why. And we don't know how all of these other players, so how RIM-A or RIM-B or cyclic IgM-P interacts with RIMK to control its behavior. So we've spent the 5 years since we published this paper really picking apart the RIM system and trying to understand how it works at a molecular level. So let's begin at the beginning. We know the one thing actually at this stage that we know regulates the RIM system seems to be being in the early stages of rhizosphere colonization. And it's downregulated in the later stages of rhizosphere colonization. So we can start there. We can say, well, we know there must be some level of expression control of the RIM system. So to look at this in more detail, we looked at expression of the RIM system using qPCR under all sorts of different conditions. And we tested a lot of different things. We grew cells in LB, and then we transferred them for 45 minutes into different conditions. So we tried things like hydrogen peroxide and salt, different pHs, and all sorts of other things that aren't shown here. And the things that we could show affected the transcription of the RIM system were transfer from nice, happy, rich LB medium at 28 degrees C to either a cold temperature, so moving to 8 degrees, or moving into this carbon-free, so starving routing solution. And these two effects seem to be additive. So what we can say at this stage is that expression of the RIM system is stimulated by nutrient limitation and by cold temperatures. So we can start to build a model for the regulation of the RIM pathway. RIM-K is controlled by low temperature and nutrient limitation. But what about control of the RIM protein itself? What about how does RIM-K function within the cell? We know we have more or less of it, but is it being affected at other levels? Well, let's have a look at another protein in the pathway, RIM-A. So RIM-A is predicted to be a phosphodiesterase, so it's predicted to degrade cyclic diGMP, and that's indeed what it does. So I should say at this stage, we've carried out co-immunoprecipitation experiments, and we can show that RIM-A interacts both with RIM-B and with RIM-K, and the whole complex interacts with the ribosome. And while it's doing this, it seems to be functioning as a phosphodiesterase. So what this means is if you take radio-labeled cyclic diGMP here, this is just a negative bovine serum albumin control, and this is a positive control where you see the degradation of cyclic diGMP to PG-PG, which is the linear form. And then you incubate this radio-labeled cyclic diGMP with RIM-A, you see the formation of this, this linear form here. So we know that RIM-A is functioning as a phosphodiesterase. And what we can also say is that RIM-A is enhancing the glutamate ligase activity of RIM-K. So if we take RIM-K and we take RIM-A and we take RPSF and we mix them all together in the presence of glutamate and ATP, what we can show is that RPSF here is modified a little bit by RIM-K under these experimental conditions. When you add a little bit of RIM-A here, you get slightly more of this RPSF is converted into the glutamated form. When you add a lot of RIM-A, the glutamated form is right up here. So it looks like what we're seeing is real stimulation of the glutamate ligase activity of RIM-K by the addition of RIM-A. And we can also see stimulation of its ATPase activity. And we've carried this out under many different conditions, and it seems to be quite robust. So that's all great. We have phosphodiesterase activity, and we have RIM-K stimulatory activity. So this is where the story begins to get confusing, because cyclic diGMP also binds to RIM-K. So we know that RIM-A binds to RIM-K, cyclic diGMP also does that. And we can show that here using this surface plasmon resonance for cyclic diGMP binding to the purified RIM-K. But cyclic diGMP also stimulates glutamate ligase activity. So if we take radio-labeled glutamate here, and we have RPSF and RIM-K in standard sort of invariant amounts, and then we add cyclic diGMP, what we can show is that more of the 14C labeled glutamate is incorporated into RPSF, which is up here. So it looks like what we have is a small molecule that's stimulating RIMK activity and a protein that's stimulating RIMK activity that degrades the small molecule. So the two things are antagonizing each other. So how can this work? It doesn't seem to make a lot of sense at this stage. Well, I'm not going to, because it's a lot of gels like this, I'm not going to go into the details of how the system functions, or I'm not going to show the evidence for it, but I will go into the details and I'll show you how we think this system is functioning. We think it's really clever. What we think is happening is that at the activity level, RIM-A at the base state is binding to RIM-K. So the two proteins work together and RIM-K activity is stimulated. At low levels of cyclic di-GMP is able to bind to RIM-K. And when it does this, it stimulates its activity even further. But then when the level of cyclic di-GMP increases further, it starts to bind to RIM-A. When it binds to RIM-A, RIM-A changes its activity from stimulating RIM-K to degrading cyclic di-GMP. So the stimulatory activity of RIM-A goes away. And then we get degradation of cyclic di-GMP. And ultimately, as the cyclic di-GMP level drops again, the RIM-A stimulation comes back. So what we ultimately have is a kind of cartoon model which looks like this. If you look at RIM-K activity here and cyclic di-GMP level abundance in the cell here, what you find is initially you will see the level of RIM-K activity go up. And then as the cyclic di-GMP level increases, the level will come down again. And RIM-A will always be trying to push things back this way. So what we actually have here is not a simple system where cyclic di-GMP increasingly activates RIM-K. We have a system where a certain concentration of cyclic di-GMP activates RIM-K, and RIM-K is turned on or off within a narrow window of cyclic di-GMP concentration. And we've worked with a computational modeler, and what we can show is that actually, using all of the parameters that we inputted into our models, this is what seems to be happening. And this also predicts that RIM-K probably functions as a tetramer with a unit of RIM-A interacting with it. So what we can see here is as the cyclic di-GMP level increases, the percentage of RIM-K which is activated within our population will increase to a certain population, to a certain concentration of cyclic di-GMP, and then will rapidly go down again. And I should say that RIM-K proteins are predicted based on the crystal structures. We haven't published this yet, but we currently predict that this protein probably functions as part of a tetramer. So this seems to be how cyclic di-GMP and RIM-A are regulating RIM-K through this sophisticated dual enzyme mechanism, which allows us to gate the activity of RIM-K within a narrow window of cyclic di-GMP concentration. So what about the other protein in the operon? What about RIM-B? Well, RIM-B, it seems to be functioning as an inhibitor of RIM-K activity on RPSF. So if we look at RIM-K activity, we have RPSF here, we have small amounts of RIM-K in our sample and increasing levels of glutamate. What we find is RPSF gets bigger and bigger. If we then add amounts of RIM-B to this, we see the successive inhibition of this activity as you add more RIM-B. So it looks like RIM-B is acting as a repressor of this RPSF activity. But something didn't sit right with us for this. Because what we have here are the ratios of RIM-B to RIM-K in each of these reactions. So we're seeing quite potent stimulation when RIM-B is present at 1/50th of the concentration of RIM-K. So, and almost complete inhibition when we're seeing, when we have 0.32 of the amount of RIM-B that we have RIM-K. So RIM-B can't possibly be interacting with RIM-K and repressing its function. Because if it were doing that, it would need to be interacting with 50 molecules of RIM-K in a huge complex. So we scratched our heads for a while. We weren't sure what was going on, but ultimately it seemed to suggest that RIMB must be having another function. So we thought, well, maybe like the thing which seems to make most sense is that RIMB might be functioning as an enzyme itself. It might be a protease. So to test this, what we did was we purified RIMB, we purified RPSF, and we purified a form of RPSF on which we had genetically added 10 glutamates at its C terminus. And this is possible because the addition of glutamates to RPSF produces a protein which is indistinguishable from a genetically encoded addition of glutamates. And we then incubated this 10 glutamate version with successively larger concentrations of RIMB. And what we saw was this. We saw that the 10 glutamate version of the protein, as the level of RIMB increases, goes down to something which looks more like this unmodified version. And when we examined this with MALDI-TOF, what we found was actually what we're seeing here is the cleavage of this protein, leaving behind about 3 to 5 glutamate residues on the C terminus. So we can add another level of regulation to our pathway. We have our activity control from cyclic diGMP and RIM-A. And what we also have now is what we're going to call discrimination control. And that's where RIM-K can add glutamates to RPSF, but those glutamates are then cleaved off by RIM-B, which is this protease. And that ultimately is going to give us an output of glutamated ribosomes. And this sort of makes sense at this stage. We can say, right, you know, we have these glutamated ribosomes. I'm not sure why at this stage we would add glutamates just to remove them, but this seems to be what the system is doing. So we can build a model. This is what we think is currently happening with plenty of question marks in different places. We think we might be seeing evidence. Certainly the E. coli protein is known to produce polyglutamate. So we think that's a possibility. We're not quite sure what these signals are, but ultimately we have kind of a nice model for how this system functions, which is obviously this part of the model is still very simple, but at this stage we kind of have an idea for how these different proteins interact with one another to mediate an output on RPSF. But this model, unfortunately, at this stage doesn't make perfect sense. And the reason for that is, if you think about this, in order for this cartoon to work, where you have one RIM system interacting with 1 or 2 ribosomes and modifying those ribosomes, you would need 1 RIM system per ribosome. And that's fine if you have 40,000 RIM systems, but what you actually have is a few hundred. So how do we make a model for how the RIM system functions if the ratio of RIM-K to RPSF, this seems to vary depending on how we do it, but we always get values in the hundreds of RPSF per RIM system. We think, and the models that we built are going on a ratio of about 1 to 500. So how does the system function? How can this all fit together? Well, we think it comes back to that thing I mentioned before about the cleavage using the RIM-B protease not being perfect. We're not seeing complete cleavage of the tail that we add. We're seeing cleavage down to about 4 glutamates. So if we have our ribosomes here, and rather than thinking of individual ribosomes, we think of the fraction of ribosomal RPSF that's either not glutamated or has lots of glutamate added to it, then we can see what effect the RIM system will have on this population. RIM-K will add glutamates and will push a small number of ribosomes towards polyglutamation. And this will be controlled by cyclic diGMP and RIM-A. But then comes RIM-B, and what RIM-B does is it pushes the population back to a state where you have 4 glutamates. And if you combine these activities together, we've actually done computer modeling of this. What you find is rather than having a small number of polyglutamated ribosomes and lots of non-glutamated ribosomes, where the polyglutamated ribosomes have one of your few RIM-K systems adding endless glutamate to the C terminus, what you end up with is a situation where RIM-B keeps cutting the population of 4-glutamates. And when you model this, what you find is if you have no RIM-B in the population, this, I should say, is a modeled population of RPSF glutamation, looking at the frequency in the population that you would have various different glutamate chain lengths with various amounts of RIM-B activity. And what you find is if you have no RIM-B present, you have lots and lots of non-glutamated ribosomes and a very small population of very glutamated ribosomes. And the more RIM-B activity you have, what you find is your population trends towards just having two different populations. And so one of them will have 4 glutamates, one will have no glutamates, but you can rewrite this as the ratio of RIM-B activity to RIM-K activity. And when you do that for a uniform amount of RIM-B activity, what you find is RIM-K activity will vary whether you have no glutamation or 4-glutamation. So ultimately what we've done here is we've transformed an analog readout. So we've transformed a situation where we're controlling RIM-K activity, and that's controlling how fast it adds glutamate to a few ribosomes, into a situation where the activity of RIM-K determines how much of your population has 4-glutamates and how much of your population has none. And so ultimately what this allows us to do is build a much more sophisticated model for how the system functions. And we published this model in 2020. We have our activity level here, which controls, based on the level of cyclic diGMP in the system, how much RIM-K activity you have. And that translates through the activity of a as yet unknown protease. We know the protease is there and we have evidence for that, but we don't know exactly what it is. What you ultimately end up with is RIM-K activity determining how many of your ribosomes have no glutamates and how many have 4. So the importance of 0 glutamates and 4 glutamates will become apparent in the next few slides. So we can go back to our original model and we can say, okay, we now know something about how the system functions, and we know something about what's happening at this level of ribosomes. We know that we're now seeing 2 populations emerging with 0 and 4 glutamates. But how does that lead to changes in our downstream functions? So how do we see interactions? Well, how does the interaction with HFQ function? And how or why does 4 glutamates matter? So to look at this, we've done a series of proteomic experiments and RiboSeq experiments, and also some quantitative examinations of levels of HFQ. And we think we have the beginnings of an answer as to what's going on at this downstream regulatory level. So first of all, what we can say is that RIM-K seems to affect HFQ abundance in a media-dependent manner. So what I mean by that is if you delete the RIM system and then you examine the amount of HFQ using an HFQ antibody, which we've done here, and this is using a WES Western blotting system. What you can see is that in poor medium, so this is an M9 par of 8 medium where the bugs are kind of struggling, what you find is the delta K mutant has less HFQ present than the wild type. And this is what we saw and what we published in our 2016 paper. If you delete the RIM system, you'll see less HFQ, and you see all of the HFQ genes change. But what was really interesting was when we then went and looked in media where we'd supplemented the medium with Cas amino acids, we saw the whole system switched into reverse. So now the delta K mutant has more HFQ than the wild type. So it looks like what's going on is that the RIM system is not so much acting to direct the activity of HFQ, it's more acting to regulate it. And it seems to be stabilizing HFQ abundance. And ultimately, this is incredibly important because HFQ has so many roles in the cell. So HFQ controls all sorts of different things. We've actually published a couple of papers looking at the HFQ regulon. This is just a review which describes it, but we also have a Frontiers in Microbiology paper in 2017, which describes how the HFQ regulon is changed if you delete HFQ. But it seems to affect all sorts of different things within the cell. I won't go into HFQ in any more detail at this stage because this is ultimately a talk about RIM-K, but suffice to say, the HFQ effect is both media dependent and incredibly pleiotropic. So how do we get at what's going on with these 4 glutamates? Why does the addition of glutamate matter? Well, to unpick this, we needed to make a series of mutants. We spent about 2 years actually just trying to come up with the conditions in which to do this experiment. And ultimately what we did was we made a RIM-BK mutant. So we now have no RIM activity and the ribosome should have no glutamation. And then we examined how ribosomal activity changed relative to the wild type. And then we made a second mutant. And the second mutant was a RIM-BK mutant, but now we'd added 4 glutamates to the C-terminus of RPSF. So now, rather than having a dynamic population, we can examine what happens when you add 4 glutamates and compare it to no glutamates, and what happens when you have nothing and compare it to wild type. And what you can see here is how every mRNA that we could detect in the cell changes depending on the addition of glutamate or the removal of the RIM system. And ultimately, this allows you to look at the HFQ effect, which is the effect of deleting the RIM system versus wild type, and to dissect from that the glutamate effect, which are where you see these handful of genes come off this red central line. So all of these genes here are affected by the RIM system. They're affected through the activity of HFQ. And so we only have a handful of genes here, but we think these are incredibly exciting genes because they're all things to do with colonizing the rhizosphere. They're things like pectate lyase and certain amino acid transporters. So it looks like what the RIM system is doing is 2 things. One of them is in a media-dependent way controlling HFQ, and the other is in a glutamation-dependent way controlling a subset of media-independent genes. So ultimately, what we have is a situation where this RIM system is controlling lots and lots of different genes within the cell. But we always had the nagging question, well, are we just looking here at a system that controls the production of ribosomes in quite a slow way? Does it just modify populations of ribosomes just to fit an environment, or are we looking at something which is a dynamic regulatory system? So how can we answer that? Well, the way that we answered that was to look at the bacterial proteome and to say, how quickly does the bacterial proteome change if you change the conditions in terms of RIM activation? So we'll go right back to our qPCR data, which shows that RIM-K expression is boosted by moving cells from nice warm LB into starving conditions and cold temperatures. And what we did now was we looked at how wild-type cells would behave in these conditions. We conducted a quantitative proteomic experiment, and we looked at how every protein in the soluble proteome changed when we plunged cells into 8 degrees. So what you can see now is by examining the ratio of different proteomes, we can say anything which moves in this direction is favored in LB at 28 degrees. Anything which moves in this direction is favored by cold temperatures and starving conditions. And then we can do the same thing looking at the RIMK mutant. So we can plot these data against each other. And what we do, well, what we find when we plot these data is that anything that falls onto a central line here, first of all, anything that's at 0, which you would expect most proteins to be, is not affected by moving into cold temperature or starving conditions. Anything that falls on a diagonal with a gradient of 1 is affected in the wild type, but is not affected by the RIM system. So the RIM knockout makes no difference to the change in the proteome. And anything which shifts away from the line, so either towards this corner or towards this corner, is affected specifically by deletion of the RIM system. And when we looked at the data, we found this. What you see here is this line of gradient 1 and significant differences, significant deviation away from the line. And what you find is this population here in green are all of the genes that we identified in our original proteome, looking at things that were controlled by the RIM system. So it looks like movement in just 45 minutes into cold, starving conditions will start the RIM system moving the entire cellular proteome away from this central axis. So it looks like what the RIM system is doing is modifying ribosomes in real time, because this is far too quick to rebuild whole ribosomes with different modification on them. What we have here is a system which is modifying existing ribosomes and changing the translational output of the cell. So ultimately, we were able to publish this model in 2020, which shows how RIM activity functions, we think. As RIM activity increases, you will see an increased fraction of RPSF glutamated. And this will ultimately affect a subpopulation of genes, possibly through direct ribosomal regulation, or possibly through interaction with other translational regulators. It will also affect, in an amino acid dependent manner, the population of HFQ in the cell. So as the amino acid concentration increases, when you have low RIM activity, you'll see more HFQ. And if you have high RIM activity, then you will see low HFQ levels, and vice versa for high levels of RIM activity. So we think it's an incredibly complex and dynamic system, but ultimately it's a system that seems to tune each ribosome in the cell and each population of ribosomes within the cell to optimally respond to the surrounding environment. And I think it's just a really nice example of how bacterial signaling really seems to function. It's not a linear pathway of one gene affects another gene affects another. It's a series of dynamic enzymes and regulators and interacting proteins that tunes how bacteria respond to their environment. So that's everything from me. Thank you very much for listening. I'd like to thank the people in my group who are responsible for this work. This is my group on the left. This work was mostly done by Dr. Lucia Grenger, who's now got her own group in France, and Dr. Richard Little. And I'd also like to thank our various collaborators and funders, and thank you very much for listening. So I will stop sharing now, and I'm very happy to answer any questions.